Okay, so here's another picture or a diagram in the PowerPoint in your textbook concerning DNA and its relationship to RNA and the different versions of RNA that are out there. So the first thing that I want you to keep in mind is that this blue kind of barrier or barricade, that is what we call the nuclear envelope. So all of this is taking place inside of the nucleus, right? And we need to get these things out of the nucleus and out here in the cytoplasm of the cell itself, right? Okay, so one of the very first things that we do is that we talk about DNA, and DNA is compact tightly inside of the nucleus, and we start to unravel that DNA and we start to replicate it. So, number one is transcription. So what we do is that we open up the DNA. Well, we don't do it. It does it itself, right? Your body knows what to do. So it opens up the DNA and we get that template strand, and that template strand creates this RNA transcript. So this is what originally is made. So all of the introns are left out. The exons are the only ones that get included. The exons are spliced together, and this creates this RNA. Okay, so from this initial RNA transcript, we begin to modify certain things. So off comes tRNA, off comes rRNA, and off comes mRNA. And the M and the R and the T all have to go out and create this factory that starts making these proteins. But it can't do that inside of the nucleus of the cell. It actually has to leave. So it's got to find some escape routes. And what it does is that it finds these channels that they basically can go through. So only the RNA can escape through these channels. Now, this nuclear envelope is made of certain things, and these things could be things like lipids, these things could be sugars, these things could be cholesterols, these things could be a number of different types of compounds. But this nuclear envelope has these outlets or these channels that allow the RNA to escape from the inside and go out into the real world where it can go and find amino acids to start to hook up. Okay, so after these RNA compounds come out, this is in the cytoplasm out here, and this is where all of the amino acids and the protein assembly begins to take place. So here is your messenger RNA, and this is relating a message on what to do. This ribosome, this is kind of your rRNA in a way, right? So this is the factory at which this whole thing begins to take part. And then your tRNAs are going out and grabbing onto these amino acids and are bringing them into the factory and sitting down on top of this RNA messenger to create this protein. And that's what you're seeing here. This is a growing polypeptide chain. That's what they call that there. So polypeptide, many peptide bonds, and peptide bonds we've talked about. We've drawn them. You should know what they look like. You know they're amino acids and carboxylic acid, or amino acids, so they have an amino group and a carboxylic acid group that are there. That kind of thing we don't have to talk about. You should know that already. So this is kind of a different view or a different perspective on what's going on with all three of these components. So a very good summary of what goes on inside of a cell and how your body makes the proteins that it needs. This could be a test question, right? It could say, describe how DNA relates to protein production. And you would have to go through and discuss each one of these steps. And you could draw that instead of writing me paragraphs if you just could replicate this drawing. So I would take a look at this picture. A picture means 1,000 words. And take a look at the picture, understand what it's trying to represent and convey. And hopefully you could kind of understand and walk yourself through this whole production process of a protein. Okay, so with that said, we can talk about this protein production. We can do some examples, we can do some synthesis, we can do some curvy arrows, but that's just something that I'm not really going to go into again. There is something, however, that I do want to talk about, and these are products that target the DNA itself. So what we're going to see is that DNA is very important to prevent the production of certain cells. And maybe there's going to be an area in your body that is producing these weird type of cells known as cancer cells. And if we could prevent that replication process from happening, then we can prevent or at least slow down this proliferation of cancerous cells in your body. So cancerous cells are kind of cells that just take off and they just do whatever they want to do, and they have no controller. They don't really have anything there to tell them you need to slow down, or you need to take a breath, or you're growing out of rate proportionately. There's no controlling aspect to them. They are very fast growing, they replicate like crazy, and when they begin to replicate, they make other cells just like them. So we get this horrible thing known as cancer because of that. So with the cancer cell, if we can take this cell, and there's some DNA in the cell, and if we can prevent that DNA from replicating, then all of a sudden we can stop the production of these cancer-causing cells. And that's very, very good news. One of the ways that we can do this is through the use of chemotherapy. And with the chemotherapy, we tend to see some radiation treatment. And the reason that radiation works so well is because it targets these fast-growing cells and it kills them immediately. And all of these slower cells take their time. So the fast-growing cells are good because those are the cancer cells that begin to multiply and divide. And that is why chemotherapy is a very good alternative to kill these cancer cells that are starting to make their presence known. At the same time, though, all cells that replicate at a very high rate get targeted. And this is why you tend to see people lose their hair, because these hair follicles and these cells that are incorporated in them, these are very fast growing cells compared to everything else. So you start to see people lose their hair because of that. You start to see people, if they stay on these treatments too long, well, their health begins to decline a little bit because of it. So it's a very good maneuver as far as short term goes. Sometimes as a last resort is what people really want the chemotherapy for. If we can treat it with some type of drug, then that's probably the best type of option. And there is a whole entire slew of drugs that are out there. They all come from natural products. Almost all of your cancer medications come from natural products, and that means basically compounds that come from plants, maybe marine organisms or microbes that are out there under the sea somewhere. So all of these cancer drugs are originating from those types of sources, and we have isolated those compounds in the laboratory. We've taken those compounds and tested them, and sure enough, they target those fast-growing cells. So that is what we kind of take a look at. If we can stop the translation and transcription and replication portions for those cells, then we can prevent your body from making these cancerous cells over and over and over and over. So what happens is that these drugs, they are typically the mycin drugs, M-Y-C-I-N. There's tons of them, so the only thing that you got to do is type in cancer mycin drug, and you're going to see probably a list of different ones that you can choose from. But these drugs interact with DNA by binding the base pairs, and we call that intercalculation or calculation. So intercalation. And these drugs bind with the base pairs inside of that DNA. And they do that by binding to the major and minor grooves. So this is one of the reasons that we talked about the major and minor grooves and why it was so important, and we mentioned it during that time because that's how some of these anti-cancer drugs work. So these anti-cancer drugs go in, they bind to these major and minor grooves, They intercalculate with the base pairs, and when they do that, it disrupts the replication process. DNA can't replicate itself anymore. And this is why these things are termed anticancer, because this stops the production of those cells from making others just like them. All right? Another thing that we got to keep in mind is that anything flat tends to give us a problem. And if you get to a point at the end of the semester where you begin to take a DNA sample and isolate DNA and run it on a gel electrophoresis and analyze it, you might be exposed to a dye called ethidium bromide. So up here at the top, I'm going to write down ethidium bromide. This is a dye that stains DNA and allows it to show up on a UV light a little bit better for us to see. You know it's bad when ethidium bromide is handled very carefully by Miss Kimberly Bufkin, because this is a compound that she is even afraid of. And this ethidium bromide is something that I don't want to get near either. So if we do work with ethidium bromide, we want to make sure that we have gloves on. We want to make sure that we have that personal protection equipment on. We want to make sure that we handle it with care, because the way that this thing works is that this compound goes onto your DNA and it sandwiches in between those base pairs and it disrupts them. And it can do that because ethidium bromide is very flat. It's kind of like a dagger. It can go in and stab and then twist. And when it stabs and twists, it disrupts the DNA for me. And this creates mutagenic kind of scenarios with the DNA itself, and this thing begins to disturb the whole way that this DNA is structured. And because of that, if you're pregnant, we advise you not to work with ethidium bromide. If you are exposed to ethidium bromide long term, you need to see a health professional at least to make sure that there's nothing that's going to go on or nothing's going to happen. But this is a mutagenic compound. This mutagenic compound can lead to cancerous compounds or cancerous cells, because what happens is that once your DNA is disrupted, it will go through and try to replicate itself, and if there's a disruption in the DNA, then it's not going to replicate itself the right way, and you could actually end up with a crazy multiplying cell because of that. The whole DNA system is completely messed up if you use these mutagenic compounds, and ethidium bromide is one of those, and that's how those work. So X-Men, yeah, there you go. Cyclops, Wolverine, the Beast, Storm, pick your favorite. So all of their DNA maybe has been mutated by compounds like ethidium bromide, where their replication point after that gave them these certain powers that they could control the weather, or they have superhuman strength, or like Magneto, raising metal pieces and slinging them across the cities. Okay, so that's the story with DNA. Replication, translation, and transcription, and a little bit about some cancer drugs as well, and why these things are so important. So we might end the lecture video talking about some example problems that we might see. And the example problems that we want to pay attention to are the example problems that relate to codons and anticodons. So if I can look at these codons and anticodons, take a look at how they start to assemble these proteins, that's probably a good enough example to do. And then we might end the video after that by talking a little bit about antiviral drugs and more medications that target this DNA, RNA, and prevent some of this replication from happening.